Good, so we're going to talk about influenza virus reassortment this morning, which, as I'm sure most of you know, is a process by which two influenza viruses that co-infect the same cell exchange gene segments. It's an important source of viral genetic diversity. To put a number on it, if two viruses differ in all 8 segments, then 256 distinct genotypes can emerge from a single cell. So we think reassortment and mechanisms of influenza virus evolution in general are important to study because this virus is a major public health and animal health concern, largely because of its rapid evolution. So you saw a schematic similar to this from Professor Klenk yesterday. This is a timeline showing the influenza A viruses that have circulated in humans over the last 100 years. You can see that the timeline is demarcated by pandemics, shown with the vertical lines. Pandemics occur when influenza A virus evolution gives rise to a strain that is antigenically highly novel and able to transmit efficiently among humans. And we know that reassortment can facilitate the emergence of pandemic strains. In particular, this would be reassortment between two viruses that circulate in different host species, which gives rise to a large shift in genotype and phenotype. In the interpandemic years, shown here with the colored arrows, seasonal epidemics recur annually when drift variants emerge that escape pre-existing immunity. We don't classically think about reassortment as contributing to this more gradual type of evolution, but over the last approximately 10 years, with the increase in availability of whole genome sequences, it's become apparent that there is a fair bit of reassortment among co-circulating seasonal influenza viruses, and that this has a major epidemiological impact. Okay, so evolution is important. How does it come about? So for influenza A viruses, there are 2 mechanisms of genetic variation underlying the evolution: mutation and reassortment. So mutation, of course, is the random introduction of errors into the genome when it's copied. The error rate of the viral polymerase is such that approximately 1 error is introduced into each genome copied. And reassortment, as I mentioned, is this exchange of intact gene segments between co-infecting viruses. So together, mutation and reassortment give rise to highly diverse viral populations within a host and within a host population. Genetic variation, of course, does not equal evolution, but under these conditions of high genetic variability, if a selection pressure or a bottleneck is applied, evolution will occur. So our overall aim in the work I'll present is to better understand reassortment at a fundamental level by addressing these 2 questions. So what underlying factors dictate the frequency of reassortment, and what are the implications of reassortment for influenza virus evolution? Before I can tell you how we are studying reassortment in the lab, I need to introduce one further concept, that of segment mismatch. So this is a major determinant of the outcome of mixed influenza virus infections in nature. I'm showing it here with 2 schematic viral gene products. So the gene products of the blue virus have co-evolved. The gene products of the red virus have co-evolved. But because of genetic drift, the equivalent gene products of these 2 viruses are likely to be divergent. For this reason, reassortant progeny that take genes from 2 distinct parental viruses tend to suffer fitness defects due to suboptimal interactions among the viral components. So as I mentioned, this is a very important feature of reassortment out in the field. But in the lab, it's very inconvenient. This is because it makes it difficult to measure reassortment in an unbiased way if the progeny viruses have differing fitness levels. So we wanted to create a system that would be free of segment mismatch, and we did so as shown here. We simply took a wild-type virus, and we introduced a small number of silent mutations into each of the 8 gene segments to act as genetic tags. We can detect these using a PCR-based method called high-resolution MELT. We call the mutative virus, or variant, or VAR for short. We also mark the 2 viruses at the protein level with differential epitope tags. So the wild-type virus has a His tag in the HA protein, the variant virus has an HA tag in the HA protein. And we use these epitope tags to track infected and co-infected cells by flow cytometry. We've made these reagents in a number of strain backgrounds now. The data I'll show you today are from these 2, so the H3N2 Panama 99 strain, and then Netherlands 60209, which is an isolate of the 2009 pandemic. So So importantly, we've shown that the changes we've made to the variant virus do not attenuate it relative to the wild type in the infection systems we use. So for this reason, we now have a system in which both parental viruses and all 254 reassortant progeny are of equivalent fitness. So this gives us a way to measure reassortment quantitatively in an unbiased way. All right, so with this system in hand, we're able to address a very simple question that had not been answered previously. What is the efficiency of reassortment in a co-infected cell? So I show you here two possible scenarios. On the left, here free mixing of two viral genomes within a co-infected cell, which would support high levels of reassortment. And then on the right, compartmentalization of the two viral life cycles within the cell, which would limit reassortment. So our data support this model on the left, free mixing. Here I'm showing you 3 genotype tables corresponding to 3 cell culture dishes of MVCK cells are co-infected with a high multiplicity of these 2 viruses, the Netherlands 09 wild type and VAER viruses. So the genotype tables, the gene segments are shown across the top of each table, and each row corresponds to a single plaque isolate from the supernatant of the co-infection. The segments derived from the wild type virus are shown with red bars, and those from the variant virus are shown with the blue bars. So we have blue all the way across. For example, this would be a variant parental genotype. And then where you see the checkerboard appearance in the middle, these are all the reassortants. Okay. So you can appreciate that we have high levels of reassortment happening here. To make it more quantitative, here I'm plotting the data against the, well, first of all, here I'm showing you a range of data points. We performed these co-infections over a wide range of MOIs. So the genotype tables I showed you correspond to these highest 3 data points performed at the highest MOI. We titrate down the amount of virus. We track the level of infection achieved by flow cytometry. So plotted on the x-axis is the percentage of cells that were infected as detected by HA positivity on the cell surface. This is either or both of the HAs that we're tracking here. Then on the y-axis is the percent of progeny viruses with any reassortant genotype. So you can appreciate that where we have high levels of infection and therefore high levels of co-infection, we see a lot of reassortment, and this of course declines as the MOI goes down. I've been included here for a reference for comparison, the solid black line, which gives a theoretical prediction for the relationship between these 2 parameters from a computational simulation of influenza A virus reassortment. So this is published, this model. The reference is given here, so I'm not going to go into detail, but it's actually very intuitive. So if you think, for example, about the situation where we have 20% of cells infected, under these conditions, according to simple Poisson statistics, of course, only a fraction of the 20% are expected to be co-infected. Nevertheless, we see 30 to 50% of progeny viruses have a reassortant genotype. In other words, 30 to 50% of viruses are coming out of co-infected cells. So clearly we see markedly higher levels of reassortment than expected in cell culture. And again, I'm not going to go into it a lot here because I don't have time, but I think it's very interesting, so I wanted to mention it. That our data suggests that this difference between the expected and observed levels of reassortment can be accounted for by high levels of incomplete genomes. Okay? So in other words, it appears that standard influenza A viruses frequently must co-infect in order to achieve productive infection, that there's a high dependence on multiplicity reactivation to yield viral progeny. Okay, so I've shown you that influenza virus reassortment is efficient. A related but separate question is a reassortment random? Okay, so we can use these genotype data to address that question. One thing we might do would be to ask whether all 256 genotypes are equally represented in our data sets. We don't have the numbers to do that properly. So an alternative approach is to do a pairwise analysis where we just consider 2 segments at a time. So if you look here, for example, at PB2 and PB1, if we just consider these 2 segments, there are 4 different possible genotypes: blue, 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 red, red, blue, and red, red. So we can ask, are these 4 genotypes equally represented? So we've done that for all possible pairwise combinations. Here we're looking only at the reassortant viruses. We exclude the parental viruses to avoid bias. I'm going to show you the results on a set of 8 scatter plots, one for each of the 8 segments. So first, before showing the data, this is the layout of those scatter plots. So if we think about PB2, for example, on the x-axis of these graphs, we're considering the viruses that have the red PB2. We're plotting the proportion of those viruses that have a red-red combination for each of the other 7 segments. Okay? So if there's a data point here at 0.6 on the x-axis, 60% of viruses have the red-red combination and 40% have the red-blue combination. On the y-axis, we consider the remainder of the viruses that have the blue PB2, and we plot there also the proportion with the homologous blue-blue combination. So when we do this, data points in different regions of the graph would indicate different types of linkages among segments. For today, you really just need to think about the middle of the graph. If reassortment is random, we would expect the data points to fall at about 0.5 on both axes. All right, so when we apply this approach to those genotype tables that I showed you, these are the results. Again, there's one scatter plot for each segment, and these other 7 segments are plotted on each. So you can clearly see that most of the data fall very close to the middle of the graph, supporting the hypothesis that reassortment between these very homologous viruses, the Netherlands wild type and variant, is indeed random. Right, so to conclude from those data, we've seen that genome mixing is efficient in co-infected cells, so we do not see evidence of compartmentalization of the viral life cycles. We've also seen that when viral genomes are highly similar, reassortment is random, which gives us an interesting insight into the viral life cycle. It suggests that the mixing of segments from co-infecting viruses occurs at one or more stages of the viral life cycle, which precede segment bundling and packaging into the viral particle. All right, we've also applied this wild-type VAERS system to evaluate the efficiency of reassortment in an intact animal host. I think this is a very important question because it's been assumed for a long time that co-infection and reassortment would be relatively rare in vivo, simply because there are a lot of target cells in an intact tissue, and it would seem unlikely that you would get 2 viruses into the same cell very often. So to address this question, we've largely used a guinea pig model. I will show you some data at the end from a ferret model, which was done with a collaboration. But in my lab, we use guinea pigs, and we use guinea pigs because they're highly permissive for human influenza virus infection. They shed these viruses at high titers from the upper respiratory tract, and human viruses transmit efficiently between guinea pigs, which allows us to include transmission in our experimental design. So to date, we've evaluated reassortment levels in guinea pigs co-infected via these 3 different routes: intranasal co-inoculation, co-infection via 2 independent transmission events, or co-infection, so a single transmission from an animal with a mixed infection. Under all these conditions, we see the viruses shed in nasal washes are up to 70% reassortant genotypes. So reassortment is clearly not rare in vivo. I'm just going to show you 2 representative data sets from these experiments. So starting with the intranasal co-inoculation, here at a high dose of 1 million PFU of each of the viruses. So under these conditions, when we use a high dose, we see that reassortment is abundant throughout the course of shedding. So that course of shedding is shown here on the right. When we use a high dose, we see rapid clearance. Already on day 4, all of the animals are at or below the limit of detection. So we're therefore looking at a short time course here of day 1, 2, and 3. And this is an array of genotype tables. So each rectangle corresponds to 1 guinea pig at 1 day post-inoculation, with the days shown at the top here and the 5 guinea pigs going down the side. Just to show you a little more clearly what you're looking at. Again, the gene segments are shown across the top, and each row is an individual viral isolate. So already on day 1 post-inoculation, we see abundant reassortment in these animals, and that sort of stays the same throughout the course of shedding. On average, overall, we see about 65% of viruses shed are reassortant. Okay. And then going to the much more sort of physiological situation where we have co-infection is achieved through 2 independent transmission events, we also see appreciable levels of reassortment here. So in this experimental design, we put 3 animals into the cage. 1 is acutely infected with the variant virus, 1 is acutely infected with the wild type, and the 3rd one is the naive exposed animal. So under these conditions, all of our naive animals quickly contract infection, but only a subset are productively co-infected. So we had 15 guinea pigs exposed in this way. 9 were productively co-infected with the wild type and VAERS, detectably so. I'm showing you the data here just from 6 of those co-infected animals. And what you're looking at is the percent reassortment again on the y-axis. These are 6 different guinea pigs. And then in the gray scale is the day post-contact with days 3, 4, 5, and 7 shown. So you can see we see appreciable levels of reassortment in all of the animals, and reassortment levels increase over time after infection. And I've included here the genotype tables that correspond to those 6 animals. So here, days 3, 4, 5, and 7 across the top, and these are the 6 guinea pigs. I include this simply to show you that where we achieve co-infection through transmission, we have a much more stochastic outcome, much more heterogeneity among the 6 guinea pigs compared to when we use intranasal inoculation to do the co-infection. Right, so we can conclude that reassortant viruses emerge readily within a co-infected host. And I think this tells us that rather than being an episodic event which occurs when conditions permit, reassortment is most likely a routine feature of influenza A virus infections in nature. Just to clarify my thinking here, the type of reassortment that would give rise to large shifts in genotype and phenotype, with human and avian influenza viruses, for example, would first require that those 2 viruses meet within the same host, which is likely a relatively rare event. However, remember that an influenza virus population, a single virus population, is actually a cloud of related variants. Okay? So reassortment among these variants will increase genetic diversity. I think that type of reassortment within a singly infected host is likely to be very important for influenza virus evolution on a larger time scale. Okay, so I've shown you that the potential for reassortment is high. I now want to tell you that a number of factors can limit reassortment levels. And these include bottlenecks at transmission or infection that would limit the genetic diversity within a host, the relative dose and fitness of co-infecting viruses, the relative timing of the 2 infections, the site of initial infection, and importantly, genetic incompatibility between divergent viruses. So I'm not going to talk about this last point because we're sticking with this wild-type VAER system to get quantitative data, but I include it because it is very important. Okay, so to show you the effects of a bottleneck, here are data we obtained when we did intranasal co-infection with a low dose of 100 PFU of each of the viruses. So this dose was found to be near the limiting infectious dose. Only 4 or 5 animals were productively infected. And among those that were infected, we saw very little diversity. Okay, so 3 of them have only the wild-type virus detected, and 1 of them largely only the VAER. So clearly, if only 1 virus gets in, then you won't detect reassortment. It's sort of an obvious point, but I think illustrates the potent effect that a bottleneck can have. Okay, we also evaluated the impact of using discordant doses of wild-type and VAER. So here we used a high dose, 1 million PFU of a total of 2 × 10⁶ PFU, and we're sampling 2 days later. So you've seen this data already, actually. When we have the 1-to-1 ratio of the 2 viruses, we see a lot of reassortment under these conditions. When we used a 1-to-7 ratio of wild-type and VAER, we saw a very different spectrum of genotypes coming out, a lot more VAER genes. However, we didn't actually have a significant reduction in reassortment levels, as you can see from the percentages shown at the right. I do, however, think that if we were to make a more extreme ratio, we would see that reduction in reassortment. Okay, so what if we don't put the viruses in at the same time, but rather introduce a time interval between the 2 infections? So we've modeled this in guinea pigs by putting the variant virus first, and then waiting 6, 12, 18, or 24 hours before introducing the wild-type virus. We then leave the animals 48 hours before sampling by nasal lavage. So here we've used a low dose, 10⁵ PFU intranasally, and under these conditions we see moderate reassortment levels at this time point. So here with a simultaneous infection, we have about 22% reassortment. We don't see a change when we introduce a 6-hour time interval. However, with a 12-hour time interval, we see reassortment levels increase, and then at later times, we see a reduction. So at 24 hours or later, no reassortment was detected, and in fact, in these animals, no wild-type virus was detected. So at the level of the whole host, at later time points, there's a superinfection exclusion that prevents reassortment. However, a short time interval will not prevent reassortment and can, in fact, increase it. Okay, so the last data set will show was obtained in a ferret model through collaboration with a group of Ron Fouchier at Erasmus Medical Center. And it was Sander Hurst and Mathilde Richard that performed these experiments to address this question. Do two viruses need to initiate infection in the same site, anatomical site, to reassort? Right, so we've compared intranasal co-inoculation to infection at approximately the same time, either intranasally or intratracheally. And here we're using our Panama 99 wild-type and VAER viruses. Again, we've also done this with the Netherlands 09, and we see similar results. All right, so first of all, the intranasal co-inoculation. Here we really wanted to see, do we see the same situation going on in ferrets as we do in guinea pigs when we do this experiment, and we do. So here, four ferrets were co-infected. We sampled on days 1, 2, 4, and 6, and you can see that we see abundant reassortment, also increasing over time, very similar to what we see in guinea pigs. These are nasal swabs, by the way. Okay, in stark contrast, when the two viruses are put in at distinct sites, we see very little mixing and very little reassortment. So in the top two animals, the variant virus was instilled intranasally and the wild-type virus intratracheally. And in the bottom two animals, it was a reverse situation with the wild-type virus intranasally. So you can see in nasal swabs that the virus that was instilled intranasally is detected almost exclusively. Similarly, in throat swabs, we detect largely the virus that was instilled in the upper respiratory tract. We do detect a little bit more of the lower respiratory virus, as seen here, but still very little reassortment. This is a very unusual sample for us. So typically, if we detect both viruses, we see reassortment. So this suggests to me that the second virus, is being transferred to the upper respiratory tract, but not actually growing there. So this strong compartmentalization of infection was surprising, because in previous work done by Sander and Mathilde, where they do a single intratracheal inoculation, they do see transit of the virus to the upper respiratory tract over time. But in the situation where we have an established infection there in the upper respiratory tract, we're not seeing that happen. So just to verify that second virus put in intratracheally did actually grow, because the data that I've shown you has not detected it well yet. Here we have tissue titers. So that's all the tissue titers are taken at the same time, of course, the time when the animal was killed. We're looking at nasal turbinate here, and then trachea and lung. And so we detect, and the colors are derived from deep sequencing of the PB2 sequence. Okay, so we genotype the viruses in these tissues by deep sequencing of just one segment. So we do see on the upper two graphs that the virus that was put in intratracheally does grow robustly in the lower respiratory tract. We see something a little strange here with ferret number 872, but we can still see that the variant virus in this case, the blue one, did in fact grow. All right, so we can conclude that dual intranasal inoculation resulted in robust reassortment in the ferret, as we have seen in guinea pigs. When infections were initiated at distinct sites, there was a strong compartmentalization of infection within the host, which limited reassortment. The implications of these findings are that dual infections in nature are more likely to result in reassortment if viruses are seeded into a similar anatomical site and have matched tissue tropisms. And so I'd like to acknowledge John Steele for his contributions. These are the members of my lab. Big thank you to Sander, Mathilde, and Ron, and the NIH for funding. Thank you for your attention.